Hello students, welcome to EPG Pathshala. I am Dr. Sanyog Jain from Naipar Mohali. So today we are going to discuss about a module entitled Challenges related to per oral delivery of proteins and peptides and strategies for their effective delivery under the paper Novel Drug Delivery System Part 2. So we will be discussing challenges and associated strategy for the transdermal delivery of the protein. Then we will discuss challenges and associated strategies for the ocular delivery of proteins, then nasal delivery of the proteins and peptides and relevant case studies. So let us start with the transdermal delivery of the proteins and peptides. So transdermal delivery applies the delivery through the skin as the delivery site. So the transdermal delivery, it is gaining considerable interest because it offers relatively large surface area of the skin available for the drug absorption, and unlike the gastrointestinal administration, it eliminates the problem of hepatic first pass metabolism and enzymatic degradation in the GIT. It also offers controlled and sustained drug delivery, and ultimately it improves the patient compliance. And dose withdrawal, it is also possible in the case of transdermal delivery. However, this transdermal delivery suffers from several drawbacks also, because each coin has two faces, like poor bioavailability, low efficiency, variability in the dosing, and skin, it is impermeable to large and hydrophilic molecules like protein and peptides. Let us see the various permeation mechanisms or route of penetration across the drug through the intact skin. So the drug molecules can either follow the transcellular route, that is the diffusion through the intact stratum corneum. So that is not possible for the protein and peptides because of their hydrophilicity as well as their large molecular weight. Other routes are possible. Transfollicular route means penetration via the trans follicle or hair follicle, so that is applicable for the nanoparticles. Besides this, there are some aqueous channels, means intercellular route, which is followed by the hydrophilic drug molecule, but again it is limited for the very small drug molecule, and the drug can also penetrate through the sweet ducts. So let's discuss what are the challenges or barrier which limits the transdermal delivery of the protein and peptides to the skin. Because these protein and peptides, these are hydrophilic macromolecules, so that's why they are not permeable across the lipophilic skin barrier. So the main barriers which limits the delivery of the protein and peptide across the skin is the stratum corneum as well as enzymatic barrier. So stratum corneum, it is the outermost layer of the skin which is consist of dead keratinized cell which is having very high lipid content. So they allow the passage of only moderately lipophilic compound which are having a small molecular weight. However, these proteins, I already told, they are hydrophilic macromolecules, so that's why they are unable to partition into the lipoidal stratum corneum, and hence they are not able to permeate across the skin. And protein and peptides being the hydrophilic macromolecule with ionic character, this is another feature, so they are totally not able to permeate through the various passive permeation mechanisms which are available for the delivery through the skin. So besides the lipoidal barrier, that is the stratum corneum, enzymatic systems also present in the skin, which is additional barrier for the transdermal delivery of the protein and peptide molecules. So there are several endopeptidase or exopeptidase, these exits. So these endopeptidase cleaves peptide bonds within the peptide structure. That example includes collagenase, elastase and fibrinolysins, while exopeptidase, these cleaves the N or C terminal of the peptide bond. Example include aminopeptidase and carboxypeptidase. Now coming to various approaches that can be utilized for the transdermal delivery of the protein and peptides. So first includes prodrug approach, that is the lipidization of the protein to make it lipophilic, chemical permeation enhancer, protease inhibitor, encapsulation in the nanocarriers, microporation, iontophoresis and sonophoresis. Prodrug approach, so that is the lipidization of the protein. So ester products, they increase the lipid solubility as well as partition coefficient of the protein and hence increase the transport of the protein across the stratum corneum. And upon reaching the epidermis, the pro-moiety is cleaved due to the enzymatic hydrolysis. So the free drug or free protein, it is released. One case study is there. Foldavari et al. Showed that the permeation of interferon alpha, it was increased by 2.5 to 5 times when delivered as an acyl derivative. They also demonstrated the dependence of the permeation on fatty acid chain length. As the alkyl chain length increased from 12 to 16, the transdermal delivery was significantly increased for the protein molecule. This figure shows the schematic diagram how the product can be synthesized for the protein molecule, and the second one shows the fold increase in the transdermal permeation of the protein in the form of prodrug. So delivery along with the chemical permeation enhancer can increase the transdermal permeation. So this chemical permeation enhancement has been widely used in assisting the transdermal delivery of the small molecules and protein and peptides as well. So various permeation enhancer that have been applied till date, they include alcohols, polyalcohols, esters, fatty acids, pyrrolidone, sulfoxides, amines, amides, surfactants and phospholipids. So the different mechanisms applied for different classes of permeation enhancer, how they improve the permeability. So for example, fatty acids act by fluidizing the stratum corneum lipid bilayer, so that's why they increase the permeability. On the other hand, alcohol act by extracting the stratum corneum lipids. Polar co-solvents such as propylene glycol, they help in the solubilizing the enhancer in the stratum corneum layer. However, there are several drawbacks associated with the chemical permeation enhancer, like their irritation potential. If they are used in the large quantity or above a certain threshold level, they cause skin irritation, which is not desirable. Second, the compatibility of the protein and peptides with the enhancer. This is again very important feature because most of the permeation enhancer, they are either surfactant or alcohol which are non denaturant for the stability of the protein and peptide, and degradation or alteration in the protein or peptide structure, it can also take place because of the permeation enhancer. I would like to present one case study on the use of permeation enhancer to increase the transdermal permeation. The effect of permeation enhancer, ethanol, and the combination of ethanol and terpene, that is cineol, on the permeation of the thyrotropin releasing hormone analog p-glu-3 methyl his proamide, in short MTRH, across the human epidermal membrane was studied. In the presence of 50% ethanol, the flux of peptide increased approximately 3 times. The donor solution of the 3% cineol in combination with 47% ethanol increased the penetration of MTRH to 1.6 microgram per centimeter square hour compared to 0.92 microgram per centimeter square hour for the TRH. So that is approximately 1.4-fold increase in the permeation. Protease inhibitor. Protease inhibitor, it is also attractive approach to improve the transdermal permeation of the protein peptide drugs. So these help in bypassing the enzymatic barrier of the skin. So the mechanism of action behind the protease inhibitor varies from case to case. So enzyme activity is inhibited by tight binding or covalent linkage of the inhibitor to active sites on the enzyme, or inhibitors may also act by chelation of the metal ions essential for the activity of the proteolytic enzymes. Encapsulation in the nanocarriers. So variety of nanocarrier system like liposomes, their modified version like ethosomes, transfersome, and nanoparticles like solid lipid nanoparticle, nanostructured lipid carrier, they have been investigated to improve the transdermal delivery of the protein molecules. So these encapsulation increase the deposition of the molecules in the stratum corneum and upper skin layer. By the virtue of encapsulation method, the delivery of both hydrophilic macromolecule, it is also possible, and additionally these nanocarrier systems, these are reported to follow the transfollicular route for skin permeation. So this is the added advantage of using the nanocarrier system. So here is one case study based on the nanoparticles for improving the transdermal permeation. Foldavari et al. Proposed lipid-based delivery system, especially the liposome, for the transdermal delivery of interferon alpha. So liposome have been found effective for the cutaneous delivery of vaccines and have shown enhanced skin delivery of the protein. Whole skin analysis showed the delivery of around 491 to 1000 nanogram of the interferon per centimeter square from various preparation, corresponding to 1,10,000 to 2,25,000 international units of interferon alpha per centimeter square of the skin. So that you can see the tremendous difference, several hundredfold difference or augmentation in the permeation can be observed. And liposome encapsulation increased the cutaneous delivery of the interferon alpha. So that is shown in this figure. Microporation or microneedle assisted delivery of the proteins. So microporation refers to the creation of the micron size channel through the skin to assist the transdermal delivery of the molecules. These microchannels bypass the stratum corneum barrier and they directly reach into the epidermal layer. So microporous channels do not reach the dermis, which comprises of the nerves. So that's why it is the painless approach for the transdermal delivery. So microneedles are generally used for the microporation, and these microneedles have been created from variety of materials like silicon, metals, glass, even polymers, and from sugars as well, such as maltose, which can dissolve when they are inserted into the skin, and their dimension varies from 300 to 900 millimeter. So microneedle mediated delivery can be achieved in different ways, like poke and patch method, coat and poke method, biodegradable polymer microneedles, as well as hollow microneedle, that is the micro injection. Here is the animation how this microneedle technology works. So these are the micron size needles which are sharp enough to penetrate the barrier, but they are small enough so that they do not reach to the nerve ending. So that's why they are painless. So when these microneedles are applied, they breach the stratum corneum barrier. They create the pore in the stratum corneum, which facilitate the transport of the protein molecule across the skin. And these pores, after some times, they are self-sealable, so they are closed, and no toxicity because of the microneedle injection it is observed. Another case studies on microneedle assisted transdermal delivery. So Lee et al. Worked on the transdermal delivery of the therapeutic antibodies using maltose microneedle. So IgG antibody was delivered under optimized condition, and the delivery was increased with increase in the concentration and length of the microneedles. Similarly, Batkar et al. Demonstrated the transdermal delivery of interferon alpha 2b in hairless rats through aqueous microchannels. So iontophoresis, this is a combined approach, microneedle as well as iontophoresis, enhance the delivery through micropores resulted in a twofold increase in the delivered dose. We have also worked on the microneedle assisted delivery of the vaccine and mediated through the chitosan nanoparticle. So, we combined the two approaches, nanocarrier based approach as well as microneedle based approach. Okay, so we investigated two different type of microneedles, hollow microneedles as well as solid microneedles. So, the native antigen delivered through the solid microneedle, it was not found very much effective, although as compared to the native antigen applied onto the skin, it significantly increased the antibody titer, but the hollow microneedle, they significantly elicited the IgG type of antibodies in the serum following administration through the hollow microneedle. Similarly, the nanoparticles applied onto the skin, they were not very much efficient in eliciting the antibody response. However, when the nanoparticle was delivered either through the hollow microneedle or through solid microneedle, they induce significantly higher antibody titer in the serum. Iontophoresis and sonophoresis, they are another strategies for improving the transdermal permeation of the small drug molecule as well as protein and peptides. So, iontophoresis, it is a non-invasive electrically assisted technique which utilize low voltage electrical current to drive the charged drug molecule through the skin. So, here the important consideration, the drug molecule should have some charge, either positive charge or negative charge. So, it utilizes the small electric current which is applied to the skin, but it is unable to create or generate shock onto the skin. For example, if you touch the mobile charger pin which is already plugged in, you won't feel any shock. So, similar kind of electrical treatment it is given to the skin, and this provides the sufficient driving force which enables the penetration of the charged molecule into the skin because of the electrical repulsion forces. Sonophoresis, it utilizes the ultrasound waves to drive the molecules into and across the skin. So, it utilizes the low frequency sonophoresis ultrasound waves, that is around 20 to 100 kilohertz, and they have been reported successful in the topical delivery of the molecules. So, this technique is also non-invasive and painless. The mechanism involved, that is the acoustic cavitation. Because of the application of the ultrasound, it leads to the formation of the micro bubble in the coupling medium, and collapse of these bubbles at the surface of the stratum corneum gives rise to the shock wave which renders the skin permeable. And another possible reason may be because of the thermal effect. Because of the application of the ultrasound wave, it generates heat. So, because of the heat, it increases the permeability of the skin. One case study on iontophoresis. So, Kartika Neri and co-workers, they studied the transdermal delivery of the insulin using iontophoresis, sonophoresis as well as microneedle alone as well as in combination. So, at the drug loading concentration of 2 mg per ml, maximum delivery was seen with the combination of microneedle and iontophoresis. Here is one case study on the sonophoresis mediated transdermal delivery. Mitra Gotri and co-workers, they reported the transdermal delivery of the insulin via this sonophoresis process. So, in normal hairless rats, the plasma insulin concentration was 101 pm. However, in diabetic hairless rat, it was below 34 pm. During the sonophoresis, the concentration of transdermally delivered insulin in the rat plasma was 77. So, that is more than 2 times higher after 30 minutes, and 178 pm, that is around 5 times higher after 1 hour. So, after transdermal delivery, we are considering the ocular route of protein and peptide for their delivery. So, ocular delivery, it is widely used to increase the efficacy and the bioavailability of the drugs that are instilled into the eye cavity, and it is used to increase their residence time on the cornea and conjunctival and corneal epithelial membrane. So, ocular delivery of the protein and peptide suffers from major drawback because of the too rapidly eliminated by the lacrimal fluid, and the drug has poor residence time in the eye cavity, so that's why the low drug contact time, and ultimately that leads to the poor ocular bioavailability. Bioavailability can be increased by reducing the effect of ocular drainage and flow on the cornea and increasing the corneal residence time. This figure shows the mechanism of ocular permeation as well as various challenges encountered after ocular administration of the drug or any protein peptide. So, instilled dose, so it is because of the rapid turnover of the tear, it is eliminated from the eye cavity. There are some metabolic enzymes present as well as protein binding can also take place, which can metabolize the drug or they can bind the drug. There is also nasolacrimal drainage which lead to the systemic absorption of the protein. So, ultimately the net ocular absorption is less than 10% of the administered dose, and secondly the capacity of the ocular cavity, it is also very much limited. Just a single drop means around 100 or 200 microliter of the formulation can be administered. So, this is another drawback. So, the ultimately after the absorption, either the drug can reach into the systemic circulation, which is not desirable, that is applied for the more than 50 or sometimes up to 100% of the drug. The ocular bioavailability is less than 10% for most of the drug, and ultimately after absorption the drug can reach into the aqueous humor and deep ocular tissues. So, different approaches like penetration enhancer, prodrug approach, nanocarrier based approach or mucoadhesive particulate carriers can be applied to improve the ocular bioavailability of the drug molecules. Similar to the transdermal delivery, penetration enhancer also plays significant role in improving or promoting the penetration of the drug through the corneal barrier. So, various permeation enhancer which are used in the ocular formation, they include cyclodextrin, dimethyl sulfoxide, ethylene diamine tetraacetic acid, sodium glycocholate and cholates, these are the bile salts, surfactants like twin 20 which is a non-ionic polysorbate, bridge 35 that is the polyoxyethylene lauryl ether, and various saponins and other bile salts. However, before using the permeation enhancer, especially in the ocular delivery, various toxicological parameters of the permeation enhancer should be thoroughly addressed before selection, because eye is very delicate organ and anything can damage to the eye which is not desirable. A scientist Yamamoto, they studied the effect of different permeation enhancer on the ocular bioavailability of the insulin. So, they concluded that the improved insulin delivery is achieved by using different permeation enhancer in the order of polyoxyethylene 9 lauryl ether, followed by sodium deoxycholate, followed by sodium glycocholate and followed by sodium taurocolate. Prodrug approach like transdermal delivery, we can also use the same concept of prodrug to increase the ocular bioavailability. So, synthesis of the prodrug can be used to change the physicochemical properties of the drug in order to improve its permeation across the cornea. So, the most common barrier in the ophthalmic drug formulation and delivery like low aqueous solubility, low lipid solubility, short retention time in the ocular cavity and systemic side effects due to low corneal and high systemic absorption, these all barriers can be overcome by the prodrug approach. Nanocarriers, so encapsulation of the drug in the nanocarrier, it not only improved the ocular bioavailability because of the improved permeation, but they also offer the sustained release. The various nanoparticles like liposome, neosomes, nanoparticle, either polymeric nanoparticle or solid lipid nanoparticle or dendrimer, and various other carriers, they have been investigated for ocular delivery. So these nanoparticles provides the protection to the encapsulated drug from enzymatic degradation, and if we can use the mucoadhesive polymer, then we can certainly increase the ocular retention time of the nanoparticle inside the corneal membrane. So here is few case studies based on the nanocarrier systems. So Plyer et al. Investigated the ability of the liposomes to deliver the immunosuppressant agent cyclosporin A to the rabbit eye. They tested the liposome encapsulated cyclosporin A or olive oil drops containing equivalent concentration of the cyclosporin A, so which are denoted by cyclosporin A liposome or cyclosporin drops. So liposome yielded significantly higher levels of the cyclosporin A in the aqueous and vitreous humor in the sclera compared to drops, evincing the requirement of the nanocarrier system. In another study, solid lipid nanoparticles were prepared using compritol, poloxamer as well as twin-80. So solid lipid nanoparticles were then investigated for cellular uptake into rabbit corneal epithelial cells and evaluated for potential cytotoxicity. And it was observed that the topical ophthalmic delivery of cyclosporin was significantly enhanced via administration of solid lipid nanoparticles. Mucoadhesive particulate carriers, because the corneal and conjunctival membrane, they have the net negative charge and hence they can interact with cationic polymers, so that can lead to mucoadhesiveness. So they interact intimately with these structures. So this ultimately result in increase in the concentration as well as residence time of the polymer associated drug. So among the mucoadhesive polymer, chitosan, which is a well known permeation enhancer and cationic charge natural or polymer, it has the unique property like biocompatibility, biodegradable backbone, and it also enhances the paracellular transport of the drug while loosening the tight junction. So another case study based on the chitosan nanoparticle using the same cyclosporin has been investigated, and after topical installation of cyclosporin loaded chitosan nanoparticle, the therapeutic concentration in the external ocular tissues, that is the cornea and conjunctiva, was found to be at least for 48 hours. After transdermal ocular, we are now going to discuss nasal delivery of the protein and peptide. So nasal route, it is another attractive non-invasive alternative to the oral route as well as invasive parenteral route. The several advantages which are associated with the nasal route, that it eliminates the gastrointestinal enzymatic degradation as well as first pass metabolism, which are the major drawback associated with the oral route. Nasal delivery, it is a needle free technique, non-invasive and painless, so obvious advantages are there. It does not require any sterile manufacturing process like which is associated with the parenteral application. It can be self-administered and facilitate the rapid onset of both local as well as systemic drug action. Attractive delivery route, nose to brain delivery, it has been investigated and it opens the door for the targeting of the drug to the central nervous system through nasal route. However, it is also associated with several drawbacks. The physical barrier of the nasal epithelium, it is the main limitation with respect to the absorption of the drug through nasal route, because rapid mucociliary clearance limits the time available for the drug absorption. So the surface area, it is very much limited, as well as residence time or transit time in the nasal cavity, it is also very much limited. So these are the two major drawback for the absorption through the nasal route, And another possible drawback is the relatively small amount of the drug that can be administered through nasal route because of the limited capacity of the nasal cavity or nostril. So hardly 200 microliter, that is just 0.2 ml of the drug, can be administered into the each nostril. So for example, if the drug dose is very high, then they are not possible to deliver through the nasal route. This figure shows the nasal epithelium and different type of cells in the nasal epithelium. So for example, there are ciliated cells, non-ciliated cells, goblet cells, gel mucus layer, sol layer, basal cell, and basement membrane. So this needs to be overcome, especially the mucus layer need to be overcome, and these ciliated cells are responsible for the mucociliary clearance of the administered drug inside the nasal cavity. Barriers to the nasal absorption. So the major obstacle in the nasal absorption is the nasal mucosa, permeation through the nasal mucosa, mucociliary clearance, as well as enzymatic degradation and sometimes interaction with the mucus layer. So like mucous membrane of the gastrointestinal tract, nasal mucosa also limits the transport of the polypeptide because they are large hydrophilic macromolecules. Mucociliary clearance, it is another barrier. So for this, the mucus layer, it is propelled from the interior towards the posterior part of the nasal cavity in a coordinated manner by the beating synchronized cilia. This is generally called the methachronal wave, which thus gradually translocates the particulates towards the nasopharynx for eventual discharge into the gastrointestinal tract. So the mucus layer is renewed every 15 to 20 minutes, which concomitantly restricts the time available for the drug absorption. So because of the mucociliary clearance, the drug molecules are absorbed or they reach into the GIT, as well as because of the mucus turnover, the residence time is hardly 15 minutes available for the absorption. The enzymatic degradation and mucus interaction, it is another important barrier. So various protease and peptidase, which naturally reduce the permeation of the various protein peptide drugs, they exist in the nasal mucosa, and other enzyme systems like cytochrome P450 family, carboxyl esterase, aldehyde dehydrogenase, epoxide hydrolase, DT diphorase, and glutathione transferase, these also exist. So in addition, the components of the mucus may undergo electrostatic interaction with the drug molecule and thereby alter its absorption and activity. So different approaches like mucoadhesive polymer, thermal polymer, pH sensitive polymers, these can be used to improve the nasal absorption of the protein and peptide drugs. Mucoadhesive polymer, they not only increase the residence time in the nasal mucosa, but at the same time they are reported to inhibit the enzymatic degradation, thereby improving the absorption of the nasally administered drug molecules. So there are few desirable properties, like they should be able to form the hydrogen bonds, and they possess the charged groups, preferably anionic groups, so that they can interact with the mucin. They should have the high molecular weight and chain flexibility and possess surface energy properties that favors spreading onto the mucus layer. Some examples of the mucoadhesive polymers like starch, cellulose, poloxamer, sodium alginate, hyaluronan, carbopol, and amylopectin. They have been investigated for the nasal delivery of the drugs. Another case study on the nasal delivery of the insulin. So insulin loaded starch mucoadhesive nanoparticles were prepared by Jen et al. And insulin loaded nanoparticles showed superior hypoglycemic action with 70% reduction of the plasma glucose and significant effect until 6 hours. In another study, Bjork et al. Showed that the nasal administration of the insulin degradable starch microsphere, they reduced significant level of the blood glucose level when compared to microsphere alone or insulin solution. Thermal and pH sensitive polymer, they form in situ gel system in the nasal cavity, consequently enhancing the residence time and nasal absorption of the therapeutic drugs. This provides a low viscosity under standard ambient condition, but they form gel upon administration into the nasal cavity as a result of either change in the pH or temperature within the local nasal mucosa. The example of pH sensitive polymer include chitosan ethylglycolic acid conjugate, which gel at physiological pH through the formation of inter and intramolecular disulfide bonds. And example of thermosensitive gel includes cross-linked chitosan and polaxamer via glutaraldehyde linking. Another case study on the in situ gel system is Ganorkar et al. Synthesized stimuli sensitive terpolymer of N-isopropyl acrylamide, which is temperature sensitive, butyl methacrylate and acrylic acid that are pH sensitive of various molecular weight to modulate the release of insulin from pH or thermosensitive polymeric beads. And it was observed that there was negligible loss of insulin at pH 2 from these beads, indicating no burst effect. At pH 7.4, insulin release was seen from all the beads and the release rate was a function of the molecular weight of the polymer. The low molecular weight polymeric beads eroded, dissolved and released most of the insulin within 2 hours at pH 7.4 and 37 degrees Celsius. The intermediate molecular weight polymeric beads swelled slightly, dissolved and released most of the insulin within 4 hours. The high molecular weight polymeric beads swelled slowly and gradually released the loaded insulin over a period of 8 hours. So students, so finally we conclude our module. There are numerous reports in the scientific literature which includes many encouraging results in the development of the viable products based on the protein and peptides. However, most of the proteins are still delivered via injectable route. However, new methods of delivery, they are appearing. For example, calcitonin nasal spray, it is already available in the market. Similarly, nasal insulin formulation, it is also reached into the market. So there is a lot of scope and hope is there for development of the newer products based on the protein and peptide, which can be available in the market. Over the coming 3-4 years, we anticipate that many more protein drugs may reach into the market.